Greetings humanity of the world. Humanity on the internet. And all participating members. We are anonymous and anonymous. There is a lot of debate as to what is the cause, or better yet, what causes autism. This video documentary is not intended to cover all of the variables which fuels autism. It is intended to provide the viewer with an understanding of the chief mechanisms related to how consumption of Monsanto's Roundup, by way of consuming genetically modified corn, soy, canola oil, sugar beets, or any other Roundup-ready crop, as well as vaccines may fuel microglial activation, a leading factor in all neurodegenerative conditions, including autism, Alzheimer's, RLS, non-situational depression, and virtually all forms of chronic illness. While this video is more directed towards the discussion of autism, the reality is that all of the factors that we will mention are unquestionably major factors of consideration in all neurodegenerative conditions. What is microglial activation, you may ask? In short, the microglia are the resident immune cells within the central nervous system. They are known to exist in two different states, and while in the surveillance mode, they play a key role in both cleaning up debris, such as dead neurons, which is another term for brain cells, in addition to fighting off infection within the nervous system. That surveillance mode role of the microglia can be shifted into an inflammatory and destructive state, where they secrete toxic substances, which is another word for glutamate, and break down the neurons. That destructive state is referred to as microglial activation. Researchers indicates that the body's ability to maintain adequate amounts of antioxidants, in particular glutathione superoxide dismutase, play key roles in determining the threshold by which the microglia may be shifted into the destructive activation state. To reiterate a previous research, the role that Monsanto plays in microglial activation is related to the fact that their genetically modified crops, in corn, soy, canola, sugar beets, along with conventionally raised wheat, that is being doused with Roundup as a drying agent, is accumulating the chemical residue within the crops that people and most conventionally raised farm animals are consuming. Research carried out by moms across America has been documenting evidence of Roundup bioaccumulation in the American population, while the USDA and the FDA have claimed that they lack the funding to test for it in the food that is being shipped across America. The argument in favor of the use of Roundup has centered around the idea that humans lack any enzymes, which can be adversely affected by glyphosate, which is the chief ingredient in Roundup. Research suggests otherwise. What is beyond dispute, however, is the fact that Roundup residue does affect the microbes living in and on our bodies, and not in a good way. Research demonstrates that glyphosate, a patented antibiotic, which is the active ingredient in Roundup, alters the microbial ecology to become dominant in pathogenic strains of bacteria, including the highly toxic Clostridium botulinum. Would you be surprised to learn that farmers and farm animals are experiencing an increased rate of Clostridium botulinum infections? You should not be. Likewise, studies of autistic populations have demonstrated altered intestinal flora, with an increase in toxic Clostridia species. What triggers microglial activation, you might ask? I will let Anonymous explain that for us.
Thank you, Anonymous. The significance of the shift of the microbial ecology to become dominant in pathogenic strains of bacteria, particularly as it relates to autism and other neurodegenerative conditions, is that pathogenic bacteria promotes an increase of two chemicals which are known to fuel microglial activation: lipopolysaccharide, or LPS for short, and interleukin-1b, or IL-1b for short. The LPS is a cell wall fragment of some of the pathogenic bacteria, and the IL-1b is one member of a class of immune system messenger molecules, referred to as cytokines, generated in response to bacterial infections. Cytokines are recognized to be prime factors in relation to how poorly we feel when we get sick. As the pathogenic bacteria grow in numbers, as they would with continued glyphosate consumption, not to mention sugar and/or refined carbohydrate consumption, they can degrade the lining of the intestines, a process called leaky gut, which then allows for the LPS to enter into the bloodstream. The immune system responds by producing IL-1b. In fact, LPS, or IL-1b, have become standard agents in studying microglial activation, so this idea that the pathogenic bacteria may fuel microglial activation by way of LPS and/or IL-1b, is well established. Additionally, it has been demonstrated that the LPS, or cytokines, produced in the periphery of the body, promotes signals up to the brain by way of the vagus nerve, causing the brain to produce these destructive cytokines within the nervous system. Both LPS and IL-1b have also been demonstrated to degrade the blood-brain barrier, which serves as an important protective fence from infections and/or other toxins which may have entered into the body. In a state of leaky gut, where the microbes from the intestines can migrate into the bloodstream, invasions from these microbes, both good and bad, may find their way into the nervous system and continually promote ongoing microglial activation once the blood-brain barrier has been compromised. Does the autistic population show evidence of LPS, or IL-1b, you may ask? Yes, studies involving autistic populations evaluating levels of cytokines, such as IL-1b, have demonstrated very clearly that the autistic population, in particular the subset with gastrointestinal complaints, have shown consistently high levels of IL-1b. In a medical article titled, Jane Neuroinflammation 2013, 1038, IL-1 sharp S, appears to be that the cytokine most involved in the quantitative traits and clinical subgroups of ASD. Involvement of IL-1 sharp S in the physiopathology of autism is generally supported by several studies reporting higher levels of this cytokine in the plasma of children with ASD, high-functioning children with ASD, and adults with severe ASD, compared with unrelated controls. It has been shown that peripheral blood cells from subjects with ASD produced higher levels of IL-1 sharp S, both at baseline and after stimulation with toll-like receptor, TLR2, or TLR4. Setting the stage for autism and an adverse event to a vaccine has a lot to do with diet, including mom's diet. Considering everything we have stated, and think for a moment of how many mothers are eating the sad diet, laden with gut ecology disrupting Roundup residue. Does a mom like to eat GMO flakes for breakfast? Perhaps she opts for a Roundup-laden whole wheat bagel, or maybe she prefers the Roundup-laden tortillas for her egg tacos in the morning. Either way, for the average mom, odds are very high that she is consuming Roundup residue with regularity. A rather common denominator to autism are mothers that have hypothyroidism, and the prime cause of hypothyroidism is autoimmunity, which has leaky gut as its prime cause. What this suggests is that mom is in a pro-inflammatory state to begin with. Her gut ecology is less than ideal, and the significance of that is that those microbes that she has living in and on her body are what are shared with her offspring, assuming she gives birth vaginally. If the baby is delivered vaginally, the baby will have the disrupted gut ecology of the mother, and have all of the basic ingredients in place for microglial activation to already be set in motion. If the baby is born via C-section, the gut ecology is not likely to be better, and may be worse. What we suspect is happening is a sort of smoldering state of low-level microglial activation in both the mom and her offspring. To make matters worse, millions of moms are feeding their babies the frankenfood cocktail called infant formula, which is commonly GMO'd soy and high fructose corn syrup based, all of which feed the wrong microbes at a critical time of brain and immune system development. As we have pointed out, autoimmunity has a common denominator in leaky gut, and autoimmune disorders are increasing at an unprecedented rate right now. Given the fact that at least 1 out of 5 of the United States population is suffering from an autoimmune condition, and many mothers do not know they have such a condition, the stage has been set for an adverse event to a vaccine. Vaccines, gas to the smoldering fire of LPS and IL-1B-driven microglial activation. As we have pointed out also, LPS and IL-1B are considered standard agents for inducing microglial activation in a lab setting, and both LPS and IL-1B are driven largely by bacterial infections. Another standard agent to consistently induce microglial activation is the cytokine interferon gamma, IFN-Y for short. A number of vaccines are live virus vaccines, and the measles, mumps, and rubella, MMR for short, is one such vaccine. Viral infections are known triggers for the production of IFN-Y. Can you see where this is going? The principal cytokine driven by the MMR vaccine has been identified as IFN-Y. If any of these live viruses from the vaccines managed to withstand an inadequate immune response, and instead became a chronic infection, which does happen, and therefore become a chronic inducer of IFN-Y, you have a perfect recipe for significant ongoing brain inflammation, particularly in the GMO-induced gut flora-altered individual, given the fact that IFN-Y is also a known promoter of leaky gut. Is IFN-Y elevated in the autistic population? Yes. It is worth pointing out that all vaccines induce some pro-inflammatory cytokine response, and are potential triggers for microglial activation. What is the bottom line, you may ask? As we have very clearly stated, the factors which promote neurodegenerative conditions such as autism can be traced back to those factors which promote microglial activation. Without question, autism, like all forms of chronic illness, are multifactorial in nature, but common threads tie many of these illnesses together, like microglial activation and leaky gut. If we can address and control these issues, and we certainly can, in most cases, we can go a long way towards preventing or reversing the collective nightmares of neurodegenerative conditions. For the sake of brevity, we have chosen not to address the issues of vaccine agent ingredients, how they promote neuroinflammatory autoimmunity, or other environmental factors, like Tylenol use, or genetic factors, such as MTHFR, which can lead to low levels of gliadin, and a ripened system for ongoing microglial activation, for life in a toxic world. In order to solve our collective health challenges, and the explosion of chronic degenerative diseases, it is imperative that we bring sanity back to our food production, and disease prevention. Doing so means eliminating the scourge on humanity that Monsanto represents, and educating the people about the necessity of detoxification strategies and nutrient repletion. We are all anonymous and anonymous by default. United as one, divided by none. We do not forgive. We do not forget.
To Monsanto and its supporters. Expect us.